All right, welcome to the ultimate challenge. Today, in this explainer, we're putting your medical knowledge to the ultimate test. We're going to be diving deep into the body's master chemical messengers with 10 objective questions in endocrinology. So grab a pen, keep score, and let's see how well you truly understand the complex network of hormones that literally keeps us functioning. Okay, let's jump right into section 1, genetics and syndromes. Here's your very first challenge. Now, we all know MEN syndromes cause tumors in multiple endocrine glands, right? But can you pinpoint the exact genetic driver behind the multi-organ tumors seen specifically in MEN1? Take a look at the options. Is it A, B, C, or D? Go ahead and lock in your answer. Well, if you chose C, you nailed it. So the crucial point to remember here is that MEN1 involves the inactivation, or essentially the loss, of a tumor suppressor gene called menin. Without this suppression, patients develop tumors in the parathyroid, pancreas, and pituitary glands. And this is fundamentally different from MEN2, which is driven by the activation of the RET proto-oncogene. It's basically the classic cellular battle of losing a brake pedal versus pressing down hard on the accelerator. Moving on to question 2. Let's see how well you know what happens when target organs just abruptly stop listening to hormonal signals. Which of these is a classic example of an acquired functional resistance? The answer is B, insulin resistance in type 2 diabetes. And honestly, this brilliantly illustrates a core concept in endocrinology. You see, while inherited genetic defects like complete androgen resistance, where a receptor mutation is literally hardwired right into the DNA, are rare and permanent, acquired resistance is a whole different ballgame. Conditions like insulin resistance in obesity or GH resistance in catabolic states are common, functional, and super dynamic. And because they're functional, they can actually often be reversed with lifestyle changes or medical intervention. Pretty amazing, right? All right, rolling right along to section 2, parathyroid and calcium. Question 3. Imagine a patient's calcium levels suddenly plummet. We know their nervous system becomes highly unstable, but which of these acute physical signs will they present with on a physical exam? And the correct answer is C. Take a look at the visual representation of Schaafstik's sign here. When calcium drops dangerously low, it actually lowers the threshold for action potentials in the nerves. This state of intense neuromuscular irritability means that just simply tapping the facial nerve causes an involuntary, dramatic facial twitch. Now, things like cataracts or Parkinsonism can definitely be associated with chronic hypocalcemia, but for acute, immediate tetany, you're looking for Schaafstik's and Trousseau signs. Okay, question 4 is a bit of a tricky conceptual one. We know primary hyperparathyroidism is fundamentally a gland problem, and secondary is a response to low calcium. But who gets tertiary hyperparathyroidism? The answer is C, and the underlying first principle here is just fascinating. In severe, long-standing kidney disease, the parathyroid glands are constantly getting stimulated to produce more parathyroid hormone to fix the calcium and phosphorus imbalances. Over time, this profound chronic stimulation causes the glands to become permanently, massively hypertrophied. So even after a successful renal transplant restores normal kidney function, these enlarged glands completely ignore the new normal and just continue to autonomously pump out excessive PTH. Let's wrap up this section with question 5. For the true genetics aficionados out there, where exactly does the blueprint error occur for DiGeorge syndrome type 1, leading to an absence of parathyroid glands? It's C22q11.21. Literally, a tiny microdeletion at this exact chromosomal locus deletes the TBX1 gene. This single missing piece prevents the proper embryonic formation of the parathyroid glands, right alongside causing thymic hypoplasia and heart malformations. It essentially strips the body of its primary calcium regulation system right from birth. Moving forward to section 3, water balance and receptors. For question 6, let's test your fundamental knowledge of how chemical signals actually travel to their targets. We've got endocrine, paracrine, and autocrine signals. So which of these uses an autocrine mechanism? If you picked D, insulin, you are absolutely right. This is such a great way to build our understanding of cellular communication. While hormones like vasopressin are your classic endocrine signals, meaning they travel far distances through the bloodstream, and gastrin acts on nearby cells as a paracrine signal, insulin is super fascinating. It operates as an autocrine signal. This means insulin doesn't just travel to distant tissues to lower blood sugar, it loops right back and acts on the very beta cells of the pancreas that produced it, perfectly auto-regulating its own factory.
Question 7 focuses on the syndrome of inappropriate antidiuresis, or SIADH. Can you identify the exact molecular cascade that forces the body to inappropriately hoard water?
The correct answer is C. The absolutely crucial mechanism driving this disease is that arginine vasopressin, or AVP, is released despite the blood already being way too dilute. This rogue AVP hits the V2 receptors in the kidneys, and that sets off a huge chain reaction, triggering a surge of cyclic AMP that violently shoves aquaporin 2 water channels directly into the kidneys' collecting ducts. The result? The kidneys just pull massive amounts of free water back into the blood, completely ignoring the body's normal osmotic rules.
All right, let's see if you could apply those exact pathophysiologic principles to actual diagnostic criteria on a lab report for question 8. Based on the Schwartz-Barter criteria for SIADH, what would you expect to see?
The answer is C, urine sodium greater than 40. Now, just think about the logic here for a second. Because the body is hoarding excess water, it slightly expands the blood volume. The body senses this extra volume and triggers what's called pressure natriuresis to try and fix it. Basically, the kidneys desperately dump sodium into the urine to try and drag some of that water out with it. So you end up with this dangerous paradox. The blood is severely diluted and low on sodium, yet the urine is highly concentrated with sodium pouring out of the body. Okay, we've made it to our final round. Section 4, Cushing's and cortisol.
Question 9. I want you to think structurally about the endocrine axis for a moment. Where does the vast majority, like nearly 70%, of endogenous cortisol overproduction actually originate?
It's B, pituitary ACTH dependent Cushing's, also known as Cushing's disease. Notice here that when a patient is making too much of their own cortisol, the primary structural problem usually isn't in the adrenal gland itself. In 70% of cases, the culprit is actually a tiny, benign tumor sitting way up in the brain's pituitary gland. This tumor just relentlessly screens at the adrenal glands by pumping out ACTH, basically forcing the adrenals to produce toxic levels of cortisol.
And here we are, your final challenge, question 10. This is a tricky piece of biochemistry regarding how cortisol actually travels through your veins to exert its effects. Which faction is actively doing the work?
The final answer is C, the roughly 5% freeform. All right, time for the final tally. How did you score? If you got this last one right, you understand a pretty profound physiological truth. 90% of your cortisol is locked up by cortisol-binding globulin, and another 5% by albumin. That literally means 95% of this super powerful stress hormone is just tightly bound, inactive cargo floating around in the blood. It really leaves us to wonder, how much of our health, our stress, and our metabolism is dictated not by the total amount of hormones we produce, but simply by the tiny 5% fraction we actually let off the leash. Think about that as you review your score, and I'll catch you in the next explainer.